Hey, my Wholemood students. So today, this is our second lecturation, saturation, studentation, and today we are breaking down organic molecules. So, the cells within you contains 4 major types of organic molecules. And before I get into this, I would like you to please hit that subscribe button, hit that share, hit that comment, and then let's get this video rolling. Okay. A whole entire mood. So, the vlogation. So today we are going to be focusing on organic molecules, and cells contain 4 types of organic molecules, namely carbohydrates, proteins, nucleic acid, and lipids. So organic molecules are really needed in the body. They are essential, and without them the body cannot function. So before we break down each of these 4 organic molecules, we're going to touch the fundamentals and the base. They lay out the foundation of organic molecules. For starters, organic molecules are molecules that contain both hydrogen and carbon, and molecules that contain hydrogen and carbon only are called hydrocarbons. All right. And then organic molecules are usually polymers, which are made up of monomers. So monomers come together to make polymers. So in simple terms, monomers are smaller subunits that join together chemically to form a bigger molecule, which is called a polymer. All right. So then, for you to get the polymer, obviously the monomers must join together, and they are joined together by dehydration synthesis. And in dehydration synthesis, it is when we are joining molecules or compounds or atoms together by, well, it's actually not atoms, molecules and compounds together by releasing water from the molecules. So basically it means that water is lost. After all, dehydration means loss of water. And this mechanism works by one molecule loses an hydroxyl group, which is an OH, and then the other molecule loses a hydrogen. So now the hydroxyl group from the one molecule and the hydrogen from the other molecule come together to form water, which is lost. And then in the spaces where the hydroxyl group and the hydrogen were lost, is where the new bond comes together and the molecules come together and form a bond. And then the opposite of dehydration synthesis is called hydrolysis, which pretty much means that we are breaking large molecules to simpler molecules by simply adding water into the molecule. So basically, when we add water, the molecule will split apart, and then the water molecule will also break apart, and then the hydroxyl group from the water molecule joins the one molecule, and then the hydrogen from the water molecule joins another molecule, and then we have 2 separate molecules. And that is how we break molecules by hydration. So dehydration synthesis and hydrolysis are pretty much the reverse of each other. With that said, now we are gonna go into our first organic molecule, which is carbohydrates. Carbohydrates are organic molecules that contain carbon, hydrogen, and oxygen in the ratio of, so it contains carbon, hydrogen, and oxygen in the ratio of 1:2:1. All right. And then now we have different types of carbohydrates, and we have simple sugars, which are pretty much simple carbohydrates. Then we have complex carbohydrates, which are made of much more longer polymers. So now we have sugars, right? And then in our sugars we have So sugars are called simple carbohydrates. Simple carbohydrates, right? Then within the simple carbohydrates, we have two kinds. We have monosaccharides and disaccharides, right? So what this means is a monosaccharide is a simple carbohydrate that contains 5 or 6 carbons. It is so simple. And then the most common simple carbohydrates that are monosaccharides are fructose and glucose, and those two are isomers, which means that they have the same molecular formula, but they have different structural formulas. So isomers are molecules that have the same molecular formula but different structural formulas, okay? And then disaccharides, by the di, it means that it's just two monosaccharides that are joined together to form a new molecule, and they are usually joined together by dehydration synthesis. Simple enough, right? And then we have complex. We have complex carbohydrates, which are obviously chains of monomers. The first one that we have is oligosaccharides. I'm sorry for my spelling. Oligosaccharides are composed of 3 to 100 monosaccharides. Okay, so now they are much bigger and much complex than monosaccharides and disaccharides. And then we have even bigger carbohydrates called polysaccharides. Polysaccharides, it just means that we have chains of hundreds, thousands, even millions of monosaccharides joined together by dehydration synthesis. Well, this includes cellulose, starch, and a whole lot of other carbohydrates. And then, so your sugars, which are simple, their function, they just provide quick energy. And then the complex carbohydrates, those ones participate in storing energy and supporting cells and organisms, okay? And then now we are gonna go into proteins. So proteins are literally the most important in a cell, the most important in your body, the most important in plant cells, the most important in animal cells, because they are the ones that do all the activities that keep your cells and you alive. Without proteins, my darling, you will be dead. So proteins are very, very much important. And then now proteins are made up of monomers called amino acids, right? So now they're made of amino acids. So amino acids are monomers that come together to make proteins, okay? And then monomers, when after monomers are made, obviously they need to join together, and then they join together by chemical bonding, and then when they bond together, they create a peptide bond. A peptide bond. So a peptide bond is the bond that holds them together and into a molecule, right? And then chains of peptides make a polypeptide, And then a polypeptide, once it falls into its functional structure, that is when we get the protein. So now we have amino acids which join together by forming a peptide bond, and then when multiple peptide bonds are together, we get a polypeptide. And then now one peptide bond is literally a single bond, and then two peptide bonds are like double bonds, three peptide bonds are like triple bonds, and then and then the structure of a protein is divided among four levels, right? We have the primary, the secondary, the tertiary, and the quaternary. So now the primary, which is also written like this, the primary structure of a protein is the amino acid sequence. So now when the amino acids are coming together, they give us the primary structure of a peptide bond. And then once the amino acid sequence is done, it goes on to the next structure, which is called the secondary structure. And then the secondary structure is the substructure with a defined shape, right? And then the shape is usually an alpha helical shape or a beta shape, right? And then after that one is done, we go on to the next one, which is the tertiary structure. And then the tertiary structure is the overall shape of the polypeptide, right? Just how it looks like just before it becomes a protein. And this is usually done, it is usually determined by the interactions between the R groups and the hydrogens, right? And one thing I didn't mention, so the structure of a protein, usually it is a carbon that is bonded to four other atoms. One of the atoms is a hydrogen. The other atom, is bonded to one other atoms or other groups of atoms. So we have a hydrogen, then we have an amino group, then we have a carboxyl group, and then we also have an R group or side chain. And then proteins have about 20 different side chains that interact in amazingly beautiful ways that gives us the different types of proteins that come together to form their functions. And then the fourth structure, which is also called the quaternary structure, is obviously the quaternary structure is the overall shape of the protein once everything has been done, how the protein looks. And then now a protein structure and shape literally determines how the protein will look and function. For starters, enzymes are different from transport proteins, which are different from other proteins. So the shape of the proteins gives us the function of the protein, and that is very, very, very important, because the shape of the protein determines the function of the protein. And then denaturing is very, very, very dangerous, because let us put it down. Denaturing is very, very significant, because this is what happens when proteins lose their function. So basically things like excessive or too much salt concentration in the cell, and too much temperature, and different a pH that is not optimum to the cell affects the proteins. And now if we have too much or too little salt concentration, too much or too little pH, or too much or too little temperature, then our proteins denature. And once proteins denature, they cannot function. And if proteins are not functioning, then you have a problem, because we need proteins to function. So now cells work so hard to keep these conditions constant. They work so hard. And then now we're gonna give enzymes a very special shout out and touch more on enzymes. So now enzymes are a chemical reaction. Not chemical reactions. Are molecules that speed up a reaction without getting used up. And then they do that by lowering the activation energy so that reactions occur at a faster rate. And then enzymes also have things called non-protein helpers, which are called coenzymes, which come together and work with the enzyme to perform the function of the enzyme. And then now an enzyme looks like this. Now, for example, so now if it looks like this, this part over here is called the active sites. The active sites is where the substrate, the reactant bind. And then the active site is very specific. It's very specific. So only substrates that have this structure can bind to this. So now the substrate is going to look a little like this. So this is the substrate. So it's going to come together and lock. It's going to get in there and lock. And then once it does that, the enzyme does its job. And then now this substrate, when it's done, it's going to leave. It's going to move from the active site and go to its function. And then it's going to be a new product. It's no longer going to be a reactant. It's going to be a product which is completely different from the reactant. And then the enzyme is still going to stay exactly the way it was. So now it just does its job, but then it doesn't change. Okay, it never gets used up in a chemical reaction. So it is a catalyst, right? And then now when you have too little, let's say you have a lot of active site concentration, and then you have little, so now you have a lot of concentration on the active sites, but then you have little concentration of the substrate. Now the reaction will occur, but not really at a slow rate. Actually, it at a slow rate. And then now when you increase the concentration of the substrate, then the concentration will cause the products to be formed at a much faster rate until optimum is reached. And then after optimum is reached, increasing the concentration of the substrates further than that is not going to do any change, because the optimum has been reached, right? Yeah. And then we also have nucleic acid. Nucleic acid are made of nucleotides. The nucleotides are the monomers that make up nucleic acids. Then a simple nucleotide usually contains a deoxyribose or a ribose. So a deoxyribose occurs in DNA and a ribose occurs in RNA, right? And then another thing that occurs is it is a phosphate. It is a phosphate group and a nitrogenous base. So now nucleotides are made up of a phosphate group, a nitrogenous base, and then a deoxyribose in DNA and a ribose in RNA. And then in RNA, then you have the nitrogenous base uracil, which is different from the nitrogenous base that is found in DNA, that is thymine. But then they also share the same other nitrogenous base, which are cytosine, guanine, and adenine. Yes, adenine. And then those are nucleic acids. And then now DNA over here. DNA is double stranded. It has thymine and it contains the genetic material. It contains the genetic material and transfers it to the next generation. While on the other hand, RNA is single-stranded. It has uracil, and it also it manufactures and synthesizes protein, right? And then one other important thing to note, that DNA is actually the one that determines the type of RNA that we have, because now we know that we have mRNA, we have tRNA, and we have rRNA. So mRNA is messenger RNA, which leaves the nucleus, and then it has codons, and then it copies the genetic material from DNA. And then tRNA is transfer RNA that carries the amino acids, and then rRNA is ribosomal RNA that is found on a ribosome, right? So now when RNA leaves the nucleus, how its structure folds itself is the one that determines whether it becomes tRNA, mRNA, or rRNA, and that usually is transcribed or described or comes from the actual DNA molecule. So the DNA is the one that determines the type of RNA that we have, or more precisely, how RNA leaves the nucleus is how it becomes the type of RNA. The last one is called lipids. So lipids are also high organic molecules, and then lipids are all very diverse, and they have one factor in common. They are hydrophobic, which means that they are repelled by water and they do not form hydrogen bonds with water. They hate water. Whenever they're in water, they're like, we don't belong here. And then now we have different kinds of lipids, and we are going to describe triglycerides, steroids, and waxes. So now triglycerides, so it's triglycerides are composed of a glycerol and three fatty acids, right? A triglycerol and 3 fatty acids. And then now we have saturated and unsaturated triglycerides. So saturated glycerides contain all the hydrogens it possibly can. And then with that said, it is saturated hydrocarbons, saturated triglyceride contains all the hydrogens it possibly can, and it is only composed of single bonds between carbons and hydrogens, and between carbon-carbon bonds, right? And then unsaturated triglycerides have at least one double bond, so that is how you separate them. And usually saturated triglycerides usually come from meat derived fatty acids, and then meat derived lipids, and then unsaturated usually come from plants derived lipids. All right, and then now we also have steroids, which are pretty much four fused rings, and they stabilize animal membranes, and they're responsible for sex hormones. And then the last one that we are going to discuss are waxes, which are pretty much fatty acids and alcohols joined together, and then they function as a waterproof, so they are waterproofers. And then now a quick recap, we have carbohydrates, which are made up of simple carbohydrates, where we have monosaccharides and disaccharides, whereby they are made of 6 carbons, and they pretty much provide a quick energy. And then we have complex, whereby we have oligosaccharides and polysaccharides, where we get compounds, things like cellulose, chitin, starch, and they provide support to the cells. They provide they store starch, and the starch which is also broken down into energy when the cell needs it. And then we have proteins, which are made up of amino acids, and then they are joined together by peptide bonds. And then when the peptide bond is done, we get a polypeptide. And then it is very important that we do not denature them, because denaturing them renders them useless. And then we have nucleic acids, which are made up of nucleotides, and a nucleotide is made up of a phosphate group, a nitrogenous base, and a deoxyribose, which is a ribose, which is found in DNA, and a ribose, which is found in RNA. DNA is double-stranded, has thymine, and contains genetic material that is transferred to the next generation. And then RNA is single-stranded, contains uracil, and is responsible for manufacturing proteins. Then we have lipids, where we get triglycerides, which have 3 fatty acids bonded to a glycerol. We also get a phospholipids, whereby it's only two fatty acids bonded to a glyceride, and then the third carbon of the glyceride is bonded to a phosphate group. And then triglycerides can be saturated and unsaturated. Saturated ones have a single bond, unsaturated have a double bond. And then we have steroids, and we also have waxes. Steroids are fused rings, and then waxes, it's acids and hydrocarbons that have alcohol. And that concludes the video today. And by the way, I am a first year student at the University of the Western Cape, and thank you very much for watching this video. I hope you learned a thing or two. The next video will be about cells, which is a very diverse topic, which is a very big topic, which can be a very complicated topic, but is a very easy topic if you do your work well and you prepare well for it. Thank you very much for watching this video. I hope you learned something from the video, and I hope that you enjoyed it. I really appreciate it. It means the world to me. See you in the next video. Thank you.